This topic, we're going to talk about enzyme inhibition. This is how do we inhibit an enzyme activity. So we talked about in other topics how we look at enzyme kinetics, how efficient an enzyme is at catalyzing a chemical reaction. We looked at how enzymes catalyze chemical reactions through mechanisms. What's the chemical basis of the rates enhancements based on the chemistry of the reaction, and used serine proteases as our detailed example. Now we're going to look at some of the ways in which we can inhibit an enzyme, and based on the inhibition, we can classify different inhibitors based on their action upon enzymes to inhibit their ability to catalyze chemical reactions. The learning outcomes are shown in this slide. So this is a summary slide of what we're going to be discussing in this topic. There are three ways in which inhibitor compounds can inhibit an enzyme reaction. It either can be done in a competitive manner, an uncompetitive manner, or a competitive manner. So there's three distinct ways, and it all depends on how this inhibitor binds to the enzyme. In the case of competitive inhibition, there are two types, and the rest there are one. So the best way to think about this is a way to write it out. Here's a way to look at it from a schematic standpoint with details. So what I'm going to do is I'm just going to focus on each figure and talk about how the inhibitor binds to the enzyme and how they're defined. So in terms of an inhibitor, we're talking about a molecule that will bind non-covalently to the enzyme. That's how we're going to first have our inhibitor based. It will bind to the enzyme in some way, okay? Now, if we had an inhibitor that was like a transition state analog, we'd expect it to bind to the active site. And so that's shown here, that the inhibitor is binding to the active site. So this gray version here is the free enzyme. In green, it's changed the conformation because it bound to the substrate and it's going to catalyze the reaction. Or it can bind to the inhibitor if it's present. And again, it binds in a little different way. Its conformation will change. So it changes a different color than gray, but it's not green, but it's still binding to the active side of the enzyme. So the gray part here is the free enzyme form, and this is the ES complex, and this is the EI complex, I being inhibitor. So for a competitive inhibitor that binds to the active site, this is the scenario we have. So the active site is either occupied with the substrate or with the inhibitor, or it's unoccupied. So these are the three different states of the enzyme, and this is called classical inhibition. The inhibitor binds to the enzyme active site. The same mode of binding of the free enzyme form could be accomplished by an inhibitor if it binds to a different site other than the active site. So say, for example, we have this version here in part B, where the active site is still this V-shaped binding pocket, but the inhibitor binding pocket is this rectangular looking feature. So the substrate binds and conformational change in the enzyme. It can catalyze reaction. This is the ES complex. This is E. And this is the EI complex here. But I is not binding to the active site. It's binding to a site on the free enzyme form that's not occupied by the substrate. So it effectively does the same thing in terms of taking out free enzyme and occupying it where the substrate can't bind to it. And so it's also competitive because it's competing with the same enzyme form as S. Both I and S are competing for this free enzyme form, both I and S in this case are competing for the same enzyme form. It's just the site of binding is different, so they call that non-classical. Okay, but it's still competitive. So both are competitive. Until you do experimental methods, you don't know where the inhibitor, if it's binding to the active site or not. Most competitive inhibitors are designed to bind to the active site because we want things to bind to the enzyme, the most tight binders, and the most tight binders will be transition state analogs, and those happen at the active site. So most of your competitive inhibition will be what we call classical, but there could be some of this other type of inhibition as well that binds to a non-active site. Okay? Imagine we have this situation where we have this extra binding site here for the inhibitor that's unique from the substrate, and we look at a different scenario. In this scenario, that the utilization of this binding site of this inhibitor not at the active site is only accomplished through a conformational change that occurs once the substrate binds. So here's your gray free enzyme form here. This is more of like a trapezoid binding site now. They're not vertical lines, so your inhibitor is not going to bind very well. Here's your substrate. It binds to form the ES complex. Now there's a conformational change that allows this inhibitor binding site to be realized through the conformational change upon substrate binding, that the inhibitor now occupies that site. So now you have an ESI complex. This ESI complex cannot catalyze the reaction because its conformational change upon inhibitor binding is such that the substrate cannot be acted upon as part of the chemical reaction. And so this takes it out of the ability to catalyze the reaction in this direction. And so this is called uncompetitive inhibition, where I binds to the ES form only. It doesn't bind to the free E form. Okay, that's uncompetitive. Non-competitive or mixed, I like calling it mixed because that makes sense to me. Non-competitive versus un is confusing. But mixed or non-competitive, it could be historical, has both. I can bind the free form and also combine the ES form. And sometimes the substrate can actually bind to the EI form, and that's shown here. But not too concerned about that. I just want you to remember that the inhibitor for non-competitive or mixed inhibition binds both the E and ES form. So there's all possible scenarios right here. So this is basically what this topic is about. How do we know, based on kinetic measurements, if we have an uncompetitive inhibitor, a non-competitive or mixed inhibitor, or a competitive inhibitor? So we can determine this based on doing enzyme kinetics, Michaelis-Menten kinetics of the enzyme and the substrate in the presence of various concentrations of the inhibitor. And we can figure out which type of inhibition we have. Do we have competitive, uncompetitive, or non-competitive? That's what we're going to be talking about. So this is showing a summary table that is looking at the effect of inhibitors on kinetic constants. So if we have a competitive or uncompetitive or non-competitive inhibitor, don't worry about the gray here. It's just the table was that, so I just left it in there. This not gray, but this peach-looking color. It's not to emphasize anything over the other types of inhibition. It's just to remember competitive binds only the free enzyme form E, uncompetitive to the ES form, and non-competitive binds to both E and ES form. So when we look at enzyme kinetics and measure the kinetic parameters, Km, Vmax, in the presence of inhibitors relative to those determined without inhibitors presence, these are the results. The Km value goes up, the Vmax doesn't change for competitive. For uncompetitive, Km and Vmax both lower, and the ratio of Vmax to Km remains unchanged. And for non-competitive, it lowers Vmax and Km remains changed. So again, I don't like memorizing things, but I like to understand things. And so although this table may be useful for you, I have to sit and pause and try to figure out myself the effect. I rather look at it in terms of graphically and think of it that way. But this may be beneficial for some of you. We're going to work with both. So this is the overall effect you see in the Lineweaver-Burke plots. And so let's look at those individually, and it may make more sense to you. Because right now when I look at this, it's like, this makes no sense whatsoever until you think about what's happening. And I like to look at it in the kinetic plots. So let's take a look at the kinetic plots for each particular type of inhibition. So this would be your competitive. Again, don't forget we got two forms, the classical, where the inhibitor binds the active site, or the inhibitor in the non-classical version binds a site that's not the active site, but it's still the free enzyme form, okay? So based on the table, this raises the Km and the Vmax doesn't change, okay? So if we were to look at a Michaelis-Menten plot shown here, here's what it would look like. So if we had our typical Michaelis-Menten curve would be this hyperbolic function. Here's your Vmax, here's your substrate concentration, no inhibitor. Minus I means no inhibitor. If we add some I, plus I, and we do the Michaelis-Menten kinetic measurements again, we would get a curve that looked like this. You see the curve kind of got reduced. The velocity isn't, initial velocities are lower in the presence of I. But if you add enough S, the Vmax will keep going up and up and up. If you add enough S, will we reach Vmax? The answer is yes, because you can see in this scenario here, either one. We'll look at the classical one. Once you form ES, this can go on to do the chemistry and form product. If I's binding here is what they call a dead-end complex. It's binding, it's releasing, it's going back and forth. It's all, you know, it's not 100% in this way. It binds, it falls off, it goes back here. And so as the reaction goes, if we add more S, we're going to change this equilibrium condition, right? Where the concentration of E is going to be, you know, this steady state condition, because we're talking about kinetics here. Steady state condition where, because we're siphoning this off, it's just not equilibrium binding. So the steady state condition would change. In other words, if we're adding more S, we're going to favor the formation of ES over EI. And so EI is going to dissociate because E is being depleted to form ES. So I is going to dissociate to try to re-establish concentrations of E. So we're having this law of mass action change these ratios. And so eventually what happens is that we add so much S that it doesn't matter how much I is there, we're going to outcompete binding to the free enzyme form by adding so much S. So we will add so much S, there's no free enzyme available for I to bind to if we add enough S. So initially the Vmax looks lower, but if you keep adding S, you'll eventually go up. So because we're competing for the same enzyme form, so we're going to outcompete the inhibitor by adding more S. And that's hence the name competitive. All right. Now with the Michaelis-Menten curves, it's hard to tell, you know, if this is a competitive situation or not. Looks, you know, if you see it go lower, it could be competitive. But a better way to look at it is to do the Lineweaver-Burke plots, and you can see here how your uninhibited reaction, as we talked about Lineweaver-Burke plots, to get 1 over Vmax in the slope. You can determine Km from it, or you can get the, you know, it's Km over Vmax, or you can get the minus 1 over Km by the y-intercept. But as you add inhibitor, you see what happenings to the lines is that they're still going through Vmax because larger substrate concentrations, we can achieve Vmax. So when we're at x is 0, S has to be infinity, so we'll be at Vmax. So you can see how Vmax can be obtained if we add enough substrate concentration in the presence of the inhibitor. But when you have more inhibitor, your Km is changing because it looks like the substrate isn't binding as well as efficiently to catalyze the reaction because some of the free E is in this form and substrate can't bind it. So it looks like it's losing the ability to bind, the enzyme's losing its ability to recognize and bind the substrate to promote the reaction. Well, that's not true. It's just decreasing the amount of E that's present. So that's why you see this value change here. Okay, so this is your competitive plot in the Michaelis-Menten with your I concentrations increasing in this direction. Okay, very distinctive. I can see this here. This is going to be harder when you look at some of the other types of plots for uncompetitive and non-competitive inhibition. If we look at our schematic, E plus S forms the ES complex E plus P. Here's your K1, K minus 1, K cat values. We went through and show how that relates to your Michaelis-Menten Lineweaver-Burke plots. Here's what you add as an inhibitor. E plus I forms the EI complex, and we can call this Ki, and that can be as a dissociation constant for the inhibitor. EI loses an I and forms E plus I. So we can think of this as a dissociation, association, binding event only, where this one is binding but catalysis as well, so it's a more complicated situation, but we can call this the Ki here. It's not Km. Km has values of K cat, K1, and K minus 1 incorporated in it. It's not a Kd. Kd would just be K1, K minus 1 with no K cat, but since this ES complex does catalyze a reaction, we have to have K cat, and that's where Km comes from. Okay, so this is a dissociation constant for the inhibitor Ki. All right, so that is the experimental way we can tell by doing these Michaelis-Menten kinetics, steady state kinetics under different concentrations of I, and looking at the Lineweaver-Burke pattern to see if it's competitive. Let's look at uncompetitive. Recall, uncompetitive is where I binds to the ES form only. So if we were to look at a Michaelis-Menten plot, we have this one. Here's I this way, without I. That's your uninhibited reaction. If you add inhibitor, everything goes down again. So it's kind of hard to tell the Michaelis-Menten plot with inhibition with uncompetitive versus competitive because it looks similar, right? Everything's lower, but the case of the uncompetitive version, you'll never get to Vmax. You cannot outcompete inhibitor binding by adding more substrate because there's always going to be this scenario. No matter how much substrate you add, I binds the ES form. So having more ES form isn't necessarily going to help you because I can always bind ES. So you're always going to have some part of the ES complex in the ESI form, so you can never hit Vmax. So you end up with a new Vmax based on how much inhibitor you have and how well it binds to the ES form. So it's kind of hard to tell from these plots if it's uncompetitive or competitive. But when you do Lineweaver-Burke plots, you get parallel lines. And why is that? Well, let's look at this scenario here. We have E plus S goes to ES, goes to E plus P. I is binding the ES form here. Again, now will be Ki that's unique for that particular binding event. Again, it's a dead-end complex. In other words, it can't do any catalysis here. It just binds or releases it. If we add more S, we make more ES, but this still happens. You can add as much S as you want. This is still going to happen. So we can never get to Vmax. Okay, there'll be some new Vmax based on how much I we add. So in the case of the first I amount, say this is I of 1 millimolar, we'll have a Vmax that's unique for that value. If we had 2 millimolar, we'd have a unique Vmax for that because we're promoting this binding. So we're taking more of this out of the catalytic step. And so we're going to lower the Vmax. Remember, it's reciprocal. So the Vmax becomes smaller, and 1 over Vmax becomes larger, so the lines go up this way. The Km here is also affected a little bit as well, as you can see, because it looks at, in terms of substrate, in terms of it being able to be recognized by the enzyme, and we're depleting ES. It's going to make it try to reestablish the ES concentration, and so you'll have more S binding to E, and so it'll make it look like the Km value has also changed. The ability for E to bind S has improved with the inhibitor, but that's just an artifact, because it's not meaning we're getting more ES formed. It's just that more ES is required because I binds to ES, and so you have to reassociate E and S to replenish that, and so it's an artifactual aspect, and the Km looks like it gets the ability for the enzyme to recognize the substrate to catalyze the reaction has improved. In other words, Km gets smaller, and that's not the effect. So if Km is smaller, then this number is going to get bigger. So there you have parallel lines. So you see parallel lines, it's uncompetitive inhibition. In terms of an inhibitor that does this, I can't think one off the top of my head that's purely uncompetitive. Now you'll see these types of plots if you're looking at 2 substrate steady state kinetics, and there's different reasons for that. But as far as an inhibitor in this case of a scenario, just binding to the ES form only, no, I can't say we have one. I'll explain why that is the case. But if you have parallel lines, you got uncompetitive. Now we'll go into the situation where we have both at play, right? So this one is non-competitive. I binds to both the free enzyme form and the ES form. And you can have interconversion here. Maybe the substrate can bind to this form or not. Again, that doesn't really come into play here. So just remember, the inhibitor binds either the free enzyme form or the ES form. If we look at the Michaelis-Menten curve for this, again, it looks very similar to the other ones. So that's why it's hard to tell, based on the Michaelis-Menten curves, whether you have competitive, uncompetitive, or non-competitive, because they all go down in the presence of the inhibitor. And it's hard to tell if they reach Vmax or not unless you go really high S. You may not go high enough. It's hard to tell, are the Kms the same? You know, there may be a little play in there. It may not be as drastic. And so let's do the Lineweaver-Burke plots. And now we see intersecting lines once again, but they're not intersecting in the y-axis. In this case, they're intersecting at the x-axis. Intersecting at the x-axis. So when you see something intersecting either in the first quadrant here or the second quadrant down here, they don't necessarily have to be at the x-axis. That is going to be non-competitive. And how can they intersect up here in the first quadrant and in the second quadrant here and on the x-axis? Well, I'll explain it in this schematic here. So here's our E plus S goes to ES to E plus P. All right. The I binds both the E form and the ES form, and maybe you have an interconversion here of EI plus S goes to ESI. All right. Again, we're not too concerned about that right now. These Ki values here, if they're equal, in other words, I binds to E and ES with the same affinity, you'll have this. The intersection would be on the x-axis. If it by one's preferentially over the other, you'll have the lines intersect either up here or down here. When they're equal binding of E and S form, it'll be here. If it likes one better than the other, it'll either be up here or up here. It doesn't matter if they're intersecting here, here, or here. It's non-competitive or mixed. It binds both free and substrate-bound enzyme forms. It will not intersect on the y-axis. If we're intersecting on the y-axis, it is going to be competitive. If it's parallel lines, it's non-competitive. So sometimes based on the affinity for the E and ES forms, there may be a little bit of ability for this to bind, say for example, to the E form. And so what happens is that your intersection doesn't occur near the Y or X axis here. It occurs way down in the floor from where you're at, on the next floor below you in your basement or the apartment below you, okay? And so when we look at these lines, they almost look parallel because the intersection point is so far away from the x-axis. So is that competitive? Or excuse me, is that non-competitive or is that uncompetitive? So when you start talking about parallel lines, how parallel are they? Are they just almost parallel but not quite? And so there may be a little binding to the E form, but mostly to the S form. And it looks like it's going to be competitive because it's 100% S form, ES form binding, but it may be only 0.1% E form binding. So that's still not 100% ES binding. So to me, that still would be mixed. So that's what I'm saying is that most of the inhibitors will either be non-competitive or mixed and competitive. The uncompetitive one would have to be uniquely only to ES. So if you have a little bit of E binding occurring, then it wouldn't be uncompetitive, right? So that's more for those who want to do enzyme kinetics and think about what's actually happening. And so if you're binding at some binding site that's distant from the active site, that site may or may not be altered very much upon S binding. If it's not altered very much, then you would have something closer, an intersecting point here. If it did alter significantly, then there wouldn't be much E binding, and so the lines would look parallel. So you get an idea for what that Ki would be versus E binding to I or ES binding to I. So what I just want you to remember is if it's intersecting over here, or there, it's non-competitive. Don't worry about these different scenarios. But I was just trying to explain why I don't know of any inhibitor being uncompetitive, because it could be explained through a non-competitive mechanism with low affinity for the E form. So if you wanted to determine that Ki, that dissociation constant of the inhibitor from the complex, we can determine that experimentally, and that's shown in this plot. So what we would do is we take whatever the inhibitor concentrations we used, and we'd need at least get 6 points here or so to get enough data to do this measurement properly. And we would have to determine what the Km value was, the apparent Km value, in the presence of I. So in the case of no I present, that would be your Km of your enzyme. And then in the presence of different I concentrations here, you have 5 different ones, you would figure out what the Km was from those individual steady state measurements, and then you would plot that as a function of Km apparent, and you should get a straight line. And so from the straight line, the equation here is y, Km apparent, equals Km times 1 plus the concentration I over Ki equals Km over Ki times I plus Km when you distribute that through that quantity there. And so now we have an equation of y equals m, which is Km over Ki, x, which is I, plus b. So b here is Km, like I said before, because 0 inhibitor would be your Km. Here's your b. Your slope is Km over Ki. And since you know what Km is, and you know what your slope is, you can find Ki. So that's how you could determine Ki for your type of inhibition pattern, by plotting those Km apparent values is a concentration of I. So the bottom line for this behavior of inhibitors on enzymes is that the Lineweaver-Burke plot will give you a good clue about what type of inhibition is occurring. Unless you have one of those things where the parallel lines aren't really necessarily parallel. And again, to prove some of this stuff, you have to do other experiments. But the Lineweaver-Burke plot is a very good indicator of if it's competitive or non-competitive in terms of the inhibition patterns based on the steady state measurements. Okay? So just to mention here, which we don't have time to go over, is if we have two substrates and we throw an inhibitor in, it gets more complicated, and so we need some other types of analysis to look at. But these inhibition plots can be important when we look at substrate binding order as well, not even with inhibitors, but to look at substrates that are which one binds first, which one's binds second, with two substrate enzymes. We can use this type of Lineweaver-Burke patterning to understand substrate binding order, but we don't have time to go over that. If the enzyme is allosterically regulated, some of the Lineweaver-Burke plots will get kind of weird. It won't be linear anymore. And in terms of some of the work and things I've done, I've seen some weird Lineweaver-Burke plots. And so you have to scratch your head thinking, well, is there something in there that's causing my enzyme to behave in a different way than I anticipated? Is it some allosteric regulation or something like that? Is there some molecule tightly bound that's causing my molecule not to bind properly, or it's not in the proper folded state? Or there's all kinds of speculations as to why you would get a non-linear Lineweaver-Burke plot. You can speculate a lot. The thing is figuring out what it is. And it's usually going to be some kind, maybe some allosteric regulation that something was so tightly bound you didn't even know it was still on your enzyme when you did the experiment. So when it works out as you think it does, it's great. And when you get some weird results, you have to think about what could it be. And there's a lot of things it could be, right? The buffer wasn't working well and the pH changed. There was some metal ions in there that interfered by binding to the protein and causing it to be in a different conformation. The metal ion was interacting with my inhibitor to cause it to be taken out of the binding to the enzyme. Many things you can speculate on. So that's what makes research hard, because you have to figure out all the details. And understanding enzyme mechanisms can take a long time, because you require a lot of different experimental evidence to support a mechanism. So I want to talk about mechanism-based inhibitors. And so you may recall this was brought up in the topic on enzyme mechanism. And although these inhibitors are designed to bind to the enzyme active site, the same site the substrate binds, they aren't considered competitive inhibitors because they are not binding non-covalently, but they are enacted upon by the enzyme and cause a covalent modification, a permanent covalent modification to the enzyme that renders it inactive. So as a result of that, they also have the name of suicide inhibitors or enzyme activated irreversible inhibitors, or KCAT inhibitors because they use the catalytic mechanism, or they're called Trojan horse inhibitors based on the Trojan horse that the Greeks used in the war they had with the city of Troy. And in order to get into the city, the Greeks and Odysseus hid into the Trojan horse. It was brought into the city, and then all of a sudden, surprise, the Greeks came out of the horse. So again, the inhibitor kind of acts like a Trojan horse in that it binds to the enzyme. In this case, the enzyme, unlike the competitive, uncompetitive, and non-competitive inhibitors, it thinks it's the substrate. It actually does some chemistry on it, and there's two fates for it once the chemistry is done on the molecule. it can either dissociate away, and then you have this activated inhibitor compound, or it modifies the enzyme and you form this dead enzyme with the covalently linked inhibitor that cannot be reversed. Okay, and so it's a very effective type of inhibition because it's very specific for an enzyme, and it kills it dead, provided this Ki is much larger than the K dissociation. You don't want this necessarily floating around and modifying something else, but it's a possibility if you want to think of it in terms of a potential pathway that could occur. It just depends on the inhibitor. The key thing is we want it to react faster with E than it dissociates, and usually that's going to be the case. That this value here is much greater than this value because, again, it's reacted on the enzyme by the enzyme, and so if it's going to modify anything, it's going to be within the enzyme active site before it can dissociate away. So these are very effective inhibitors, very specific, and it takes the enzyme out of any action that it can promote, whereas non-covalent inhibitors can be outcompeted, for example, in the active site, whereas these modify the active site so the enzyme is dead. So if you were to design a drug, you'd want a drug to be like this. A mechanism-based inhibitor would be your best drug molecules. But they're hard to design. So let's look at a couple examples, though. Consider the acetylcholine esterase, which is an enzyme that hydrolyzes acetylcholine. So acetylcholine is a neurotransmitter that's released between two adjacent nerve cells, and this is what is released to propagate the nerve impulse from the presynaptic nerve to the postsynaptic nerve through the synaptic cleft. These acetyl-CoA molecules exist in little vesicles, and they will fuse with the membrane and then be released into the synaptic cleft to bind the acetylcholine receptor and propagate the nerve signal. If left unchecked, the nerve just keeps going, so we have to shut this signal off. And so what's released is this acetylcholine esterase, which will hydrolyze the ester here. So you're basically having water attacking this carbonyl to break up this carboxylic acid ester, and hydrolyzing it to acetate and choline, and so therefore you shut the signal off. Okay, so that's the function of acetylcholine esterase, is to shut off the signal when appropriate. Here's an example of a mechanism-based inhibitor or reversible inhibitor of acetylcholine esterase. So if we look at the active site of acetylcholine esterase, it has a serine in its active site, and the role of that serine is very similar to the role of serine in the serine proteases. It acts as a nucleophile to attack the carbonyl carbon of the ester to promote ester hydrolysis. So it's exactly the same type of mechanism. You have to have abstraction of this proton by a neighboring base in the enzyme active site to promote the nucleophilicity of this alkoxide that's formed that will attack, in this case, the phosphate, the phosphorus atom of this diisopropylfluorophosphate. And this fluorine is a very good leaving group as well. And so basically you have a displacement of the fluoride ion and covalent attachment of the serine oxygen to the phosphorus group, the phosphorus atom, to generate this covalently linked enzyme that is no longer functional. So basically what you've done, you've alkylated your serine hydroxyl, and it's no longer enzymatically active. So chemical weapons like sarin gas have a similar mode of action on acetylcholine esterase. You basically modify the enzyme, it's no longer active. So if you don't have active acetylcholine esterase, nerve propagation cannot be shut off. So muscles will continue to contract even though they don't need to be contracting, and that's going to cause all kinds of issues. Paralysis could result, and since you can also affect respiratory processes and muscles there, you can't breathe anymore and you die. So that's the biochemical basis of its toxicity, is that these compounds modify acetylcholine esterase in a mechanism-based fashion to render the enzyme inactive. Could this molecule be used to inhibit serine proteases? Yes. And so this can also be an effective molecule to use for inactivating proteases. And there's some derivatives of this that are actually used, and I've used them before as well. Very effective molecules for inhibiting enzyme like proteases or esterases, because they have a similar mode of action through activation of the serine hydroxyl group to be nucleophilic through proton abstraction. The last section I wanted to talk about was in terms of some of the regulatory schemes in enzyme inhibition. So when we're talking about enzymes, we're not usually talking about them by themselves, but in a cell in relation to other enzymes as part of a pathway. And as a result, their regulation is important for the flux in a metabolic pathway. So several regulatory schemes are used to increase or decrease a specific enzyme in a pathway. And we'll be talking about these in detail when we get to metabolism. But right now I just wanted to show some examples and a couple of the ones that you should know, because you see these a lot in metabolism. So one example would be shown here. We have substrate A, the product B, which then gets converted by another enzyme to C, and another enzyme converts C to D. And so this is kind of a linear pathway here, with each arrow being catalyzed by a different enzyme. Simple end product inhibition is basically the accumulation of the metabolite at the end of that pathway. If the concentration is high enough, will bind to the enzyme in the first step and thus slow down the generation of D, because we don't need D anymore. We got enough D. Concentration builds up to too much. It now starts to bind to the enzyme. Okay, it could be a non-competitive inhibitor. It could be a competitive inhibitor. More likely non-competitive, since it's kind of removed from substrate A in terms of other modifications. But it can bind to the enzyme and prevent A from being effectively catalyzed to form B and slow it down. So this is a very effective way to slow down the production of intermediates in a long metabolic pathway, because the end product will inhibit the first step. So you basically shut off everything else, because if you slow down production of B, you also slow down the production of C. So it's a very effective way, this feedback inhibition. And we show inhibition by a line indicating where the enzyme is going to be effective in its blunt end. So here are some examples of more complex inhibition patterns. They have different names. I don't want you to memorize the names. I don't want you to memorize the patterns. I just want you to appreciate the complexity involved. So in the case of A eventually in a pathway to form D, sometimes there's just not one enzyme ready to use D, but there could be two enzymes. And so the different enzymes in this branching point here are controlled by feedback inhibition from different products in the pathways that are being promoted through the use of D. You can have multiple inhibition of a particular product in a pathway. For example, E may inhibit its production through the enzyme from D, but it also can affect D by inhibiting A, for example. So you get this effect of E being an inhibitor of two different enzymes. All right. So it can get more complicated. It can get more complicated. And these are just some simple examples. You can imagine in a cell, right? You have many pathways and branching off points, and concentrations are changing based on cellular requirements. And so you can imagine a lot of feedback inhibition patterns that can be determined. So today's technology, we're able to measure a lot of metabolites through metabolic analysis through mass spectrometry and quantify their changes over time. We're also able to measure protein concentrations through mass spectrometry analysis over time courses, as well as their modifications, and when those modifications occur that are involved in their regulation, as well as gene expression sequencing tools that we have available. So you can look at a system-wide analysis of how a cell responds to a particular environmental stressor or drug, or comparing disease versus healthy cells, cancer versus non-cancerous tissue, and figure out what's going on there in a biochemical level. There's also activation, and activation turns on a particular enzyme or activates it further. The one you need you see a lot and you should be aware of is substrate activation. That's where the accumulation of A at high enough concentration will induce the activation to form more B, and to activate that pathway, which will eventually increase the concentration of D. But if it had some feedback inhibition, if you get D too high, it would then start to inhibit that reaction to slow it down. So you can imagine different combinations of activation and inactivation. Again, with this substrate activation, it's going to be mostly through multi-subunit enzymes that promote the higher concentrations of A, better binding of A, and more production of B, and eventually D. So a lot of these allosteric regulators go through the enzymes that have multiple subunits. They could still be monomeric, but often multimeric enzymes have multiple binding sites and allow this allosteric regulation to occur through the binding of metabolites and regulation conformational changes that relate to substrate binding and catalysis. Here's an example of more complex activation and inhibition patterns. So here's another activation pattern. If you bring D high enough in concentration, one way to change the concentration of A to slow down the production of D is to activate another enzyme to convert A into something else. Instead of converting it to B to make more D, you can activate another enzyme to have A be converted to E. That will slow down the production of D. A combination of activation with the arrows and inhibition with the blood ends are shown here. So again, these can be quite complex, and you just need to know basic end product inhibition and substrate activation. And the rest of these complexities, you'll see them kind of unfolded when we study the metabolic pathways later on in other topics in this course. So for this topic, focus on the inhibition patterns: competitive, non-competitive, uncompetitive. Can you recognize those from Lineweaver-Burke plots? Do you understand where the kinetic parameters are in those plots? Can you understand what I is binding to in a competitive versus uncompetitive versus non-competitive situation? And that's what I want you to think about, as well as what's a mechanism-based inhibitor and how that differs from the non-covalent inhibition seen with competitive, uncompetitive, and non-competitive. I provided some problem sets that have data that you can plot and some different scenarios to think about inhibitors and how it relates to drug design and the understanding of the role of controlling enzyme activity through small molecule binding, be it inhibitors or in the case of activators. So work on the problem sets, those in the mastering assignments, and read the sections in the textbook to get a better understanding of looking at enzyme inhibition.